In this lecture, we're going to talk about blood. Okay, so what's in blood? Well, blood is really made up of fluid and cells. Okay, and the fluid contains components that are dissolved in it, molecules, and gases. Those molecules may be, for example, wastes, nutrients, and gases such as carbon dioxide and oxygen. And the blood is going to travel in blood vessels to all the tissues. Okay, and so realize that blood also is transferring heat around the body, for example, to the surface of the body when you get very hot, and the surface will then cool the blood. And the blood also carries infection-fighting components that we will talk about in this lecture. Let's focus now on the fluid of the blood. Another name for the fluid of the blood is plasma. And plasma is really about 90% water, but it also contains things that came through digestion, such as vitamins and amino acids, glucose. And blood also contains hormones, which are chemical messengers that are passed from one organ to another to maintain homeostasis. As well, cells give off waste products, which are converted to urea, that are then carried by the blood to the kidney, and then the kidney is going to excrete waste as urine. The blood also contains proteins called albumins, and the albumins are formed in the liver. And the primary purpose of albumins, which are made in the liver, are to keep water in the blood. So they maintain what is called an osmotic pressure of blood and keeps the blood pressure up. And then as well, you have proteins in the blood that are called blood clotting factors that participate in the formation of clots and making a scab. And then there are antibodies in the blood, which are large proteins that fight infection. We'll look at those a little bit later. Let's transfer our time to the blood cells, okay? And so specifically red blood cells we're talking about on this slide. Red blood cells are circular, but they also have two concave sides. So they kind of go in like a donut, but the hole doesn't go all the way through. And if we look at a side view of a red blood cell, we can see that they're actually very thin. And this allows red blood cells to fit through capillaries, which are the smallest of blood vessels, where the exchange of oxygen and nutrients occurs into tissues. So red blood cells are formed originally in the bone marrow. And they only live for about 120 days, so about 3 months. So they have a really short lifespan compared to other cell types. And the reason for this is that red blood cells, as they develop, they actually eject their organelles. And so, for example, their nucleus and their mitochondria all go out. And these are all the organelles are what are required to maintain the life of a cell. But the red blood cells no longer contain those organelles. And at that point, they are considered to be erythrocytes or mature red blood cells. And the erythrocyte only contains hemoglobin. Okay, and so you can think of erythrocytes as packages of this molecule called hemoglobin. And hemoglobin is the protein that binds to oxygen and carries oxygen in red blood cells. And so red blood cells, the mature erythrocyte, can really be considered to be simply a package of hemoglobin whose purpose is to carry oxygen to the body tissues. Hemoglobin can also bind to carbon dioxide and is one of the ways that carbon dioxide leaves the blood or is carried to the lungs to be exhaled. Okay, so let's talk next about the white blood cells. Okay, so blood also contains white blood cells, and these are called leukocytes. And the primary purpose of a leukocyte is to help defend the body against pathogens. A pathogen is a disease-causing agent, so these white blood cells are protecting us. And they also travel in blood vessels, just like red blood cells, but they also travel in lymphatic vessels. And interestingly, they can squeeze out of these vessels into the tissue spaces where they can attack pathogens that are in the tissues. Unlike red blood cells, you actually have many different types of white blood cells, and we're not going to go into all of them here. But realize that some of them actually live for many years, and some of them are short-lived. And so let's look at some examples of white blood cells. On this slide, we're just going to talk about two examples. And one example is the macrophages. These are very large cells. I'm going to draw a picture of one. And macro means large, and phage means it carries out phagocytosis. Remember, cell eating. So these are cells that protect our bodies by eating pathogens. And remember that process of endocytosis, engulfing a solid particle, is called phagocytosis. There you have a yellow bacterial cell. And the giant purple macrophage is swallowing up the bacterial cell and will digest its components in the lysosome of the macrophage. So that's one type of white blood cell. Second type of white blood cell is called the B cell. And B cells are specialized to secrete antibodies. These are large proteins that are going to bind to foreign invaders. They're actually Y-shaped proteins, and they glom on to foreign invaders or pathogens. And they actually form complexes with pathogens, especially small molecules that get into the body. If an antibody forms a complex with a small molecule, then it becomes large enough to be recognized by macrophages. So in this way, antibodies protect us from foreign invaders. Another component of blood is called the platelets. Platelets are interesting because they're actually fragments of thrombocytes. Okay, and so these thrombocytes are cells, and the cells along their development become fragmented, and they're actually cell parts that look like plates, hence the term platelets or little plates. And they really only live for 7 to 11 days. They're very short-lived, and that's because their requirement is for blood clotting. And so what happens when you cut yourself is that if you cut into a blood vessel, the platelets form what's called a platelet plug at the damaged site of the blood vessel. I'll attempt to draw that here. And so the platelets aggregate to the endothelial lining of the blood vessel, and they form a little plug. And then that attracts additional blood clotting factors or proteins will arrive at the scene. And they are coming out of platelets in most cases. And then this leads to the formation of a protein called fibrin, which is a sticky protein that leads to the formation of a scab ultimately. So there are 3 different alleles for blood types in our population, and that leads to 4 different types of blood. Let's talk about those blood types on this slide. A person can either be type A, can have type B blood, could also have type AB blood, or could have type O blood. And that has to do with the surfaces of red blood cells. In type A blood, I'm drawing these cell surface molecules we call antigens. Okay, and the term antigen really refers to any molecule that activates the body's immune system. I'm writing that at the top of the slide. Any molecule that activates the body's immune system is called an antigen. And of course, if you have type A blood and you have the type A antigens, they're not going to attract or activate your immune system because they're considered to be self. Okay, but if that blood gets transferred into somebody else's body, then the type A antigens will activate that body's immune system. And so type A blood has a certain set of red blood cell surface markers called antigens. And then type B blood has a different set of cell surface antigens. If a person has type AB blood, then they have both types of antigens on their red blood cell surfaces. And a person who has type O blood doesn't have those antigens on its cell surface. Okay, so let's look at what else is in the blood of each of these people. Okay, so interestingly, a person who has type A blood will naturally have what are called anti-B antibodies. Okay, and that means that they have antibodies against type B blood. And so if a person with type A blood receives a transfusion of type B blood, those anti-B antibodies are going to react with the transfused blood, and that could cause what is known as clumping or agglutination in the blood vessels of that person. The person could die. They received the wrong blood in a transfusion. Okay, now so likewise in type B blood has anti-A antibodies, which means that blood has antibodies against type A blood. So a person with type B blood receives a transfusion of type A blood. Likewise, they could die because their blood will clump. And type AB blood does not have antibodies against either anti-A or anti-B antigens, okay? And so that's because both the A antigens and the B antigens are recognized by the immune system as self molecules. So the immune system is not going to attack its own molecules. And then a person who has type O blood is going to have antibodies against both A antigens and B antigens. So a person with type O blood has anti-A antibodies and anti-B antibodies. Okay, so because a person with type AB blood does not have antibodies to either of the antigens, it's called the universal recipient, meaning in a transfusion it could take any of the other blood types in an emergency situation. A person with type O blood can only receive type O blood, but because its cells do not have antigens, it is often called the universal donor. Let's look at how blood type is determined. So a person comes into the lab and blood is drawn and they have an unknown type of blood. And Well, what's going to happen if they have an unknown type of blood is that the medical staff is going to determine whether that blood causes a clumping or agglutination reaction when it is mixed with anti-A or anti-B antibodies. Okay, so if it is recognized by anti-A antibodies, then it's going to cause clumping. Okay, and that would mean it has type A in it. It would either be type A blood or it could be type AB. But we know that if it's going to cause clumping with anti-A antibodies, then it has to have the type A antigen in it. And likewise, if it causes clumping when anti-B antibodies are added to the blood, then the unknown blood sample must have the B antigen in it. It could either be type B blood or it could be type AB blood. Okay, so let's look at an experiment here where we have an unknown type of blood. And we're just going to take a little bit of this blood and put it on a microscope slide. And then we're going to add anti-A antibodies or anti-B antibodies. So we take a little bit of blood, let's put it on a microscope slide here. It's a microscope slide, and we add anti-A antibodies to one microscope slide, anti-B antibodies to the other microscope slide. And in the first case, we're going to put a drop of blood onto the microscope slide. And in the second case, we're going to put a drop of blood. We're going to look at the results. And what we find with the anti-A antibody, blood looks pretty normal. Okay, we're not seeing any agglutination when we look through the microscope, which is not shown here. Okay, so if antibodies against type A don't cause a clumping reaction, then this blood must not contain a type A antigen. Okay, so we've ruled out that this blood could not be type A blood. And could it be type AB blood though? No, it could not be type AB blood either because it does not contain the type A antigen. Let's look what happens when we treat this blood with antibodies against type B antigen. Okay, now we're seeing the clumping reaction, which is also called agglutination. Okay, so we can conclude from this experiment that the blood is type B. How do the genetics of blood typing work? So realize that it is a multi-allele system. You have more than 2 alleles here. You've got actually 3 alleles in a population. So amongst all of us, there are the A, B, and O alleles. But an individual only has 2 alleles, right? Because we have 2 copies of each chromosome. Okay, so what are the possibilities for what individuals can be in our population? An individual could have these genotypes, AA or AO, BB or BO. Okay, so this is talking about B on one chromosome and B on the other, or B on one chromosome and O on the other. Person could have AB, right? An A on one chromosome and a B on the other, or it could have 2 copies of O. If a person has A and B, they're considered to be codominant. Okay, that means that both are expressed. Okay, and you have to remember back to the blood cell of a person with type AB blood. Remember that both antigens were on the surfaces of those red blood cells. Okay, and then in the genetics, type O is considered to be recessive. Okay, recessive means it is masked by a dominant. Okay, and so it's not going to show if there's another gene on the other chromosome that is either A or B. Okay, so in the first case of AA and AO, that person would have type A blood. BB and BO, that person would have type B blood. A person with AB would have type AB blood, and a person with OO would have type O blood. Let's do a little genetics problem and see if you can figure this one out. Okay, so Mary has type A blood, but her father has type O. Okay, so you have to think about what's the genotype of Mary. And let's say that you have somebody named Jack. Okay, so her partner. And Jack has type B blood, but his mother has type O. So you have to think about what's Jack's genotype. Okay, so if Mary and Jack have children, what are the chances that their children are going to have, let's say, each blood type? How do you figure that out? Okay, so what are you going to do first? So you have to go back and remember your genetics. Okay, so the first thing you do is you have to figure out the genotypes of the two parents. So you have to figure out Mary's genotype. So if she has type A blood, could she be AA? No, she couldn't be AA because she's got an O from her dad. So Mary's genotype is going to be AO. And likewise is actually true with Jack. He can't be BB because he's got an O from his mom. Okay, so Jack is going to be genotype BO. Okay, what are we going to do with this information? We're going to make a Punnett square. Okay, so we put Jack on one side and Mary on the other, and then we're matching up each of these gametes, and look at all the different types of blood that their children could have. Okay, so we've got a big answer evolving here. And so we've got what is the answer? So 25% could have AB, right? 25% chance that their children will have AB blood. 25% chance that their children will have type BO, okay, which is really type B blood. And 25% chance of AO, which is type A blood, and 25% chance that they will have a child with type O blood. Okay, and then the top one is 25% chance of a child with AB blood. Okay, so they have 25% chance of having a child with all of these different blood types. So hopefully you arrived at that answer. Then people always say, well, how come I hear somebody say that they are A positive or A negative, right? What's that about? Okay, so when you hear positive or negative, you're hearing them talk about another type of antigen that is found on the surface of red blood cells. And that antigen is called the Rh factor. Okay, so in the next slide, which is actually our last slide, we're going to talk about the Rh system. Okay, an Rh system gets its name because it was originally discovered in the rhesus monkey. There's a little picture of the rhesus monkey. And so people are either positive or negative. In this case, it's not a multi-allele system. You're either one or the other. And so typical genetics apply. And again, it refers to the antigens found on red blood cells. And why is this an issue? Well, it can become an issue if a woman gets pregnant. Okay, and so let's take a look at what happens in pregnancy. Okay, so the issue is mom gets pregnant and she is Rh negative, but let's say the fetus is Rh positive. Okay, so she goes through her normal pregnancy, no problems, and then there is the delivery, and everybody's happy, okay? No problem there. Okay, but the thing about deliveries is that there's often can be a tearing of tissue, and that results in a mixing of the blood. Okay, so that could happen even though the baby's out and the baby's normal. No worries. Okay, but then could be that that mom decides she's going to have second child. So along comes pregnancy number 2. And the mom is still Rh negative. But the thing about the mom at this point is that at that first delivery, she her blood ended up coming into contact with the blood of the fetus. So she would have made antibodies against the Rh factor in that first child's blood. So she now contains antibodies against Rh. Okay, but now she's got her second pregnancy, she's got her second fetus, and the fetus is Rh+. So those antibodies that she now has are going to reject that fetus. So that can be really dangerous. Mom's body rejects fetus, condition called erythroblastosis fetalis. But fortunately, modern medicine has stepped in, and there's a treatment for that, which is to give the mom, pregnant mom, anti-RH. So basically a medication that sops up all of the anti-RH antibodies so that the mom then is not going to, her antibodies are not going to be able to react with the fetus and reject the fetus. So in the end, the baby is delivered and she has a second normal child, and everybody lives happily ever after. And on that note, I think we will end our lecture on blood. Thank you for listening.